The previous two videos have focused on the fact that proteins are polymers of amino acids, and 20 different amino acids are used to build proteins. Well, in order to build the polymer, these amino acids have to be joined to one another, and the way they're joined is through what are called peptide bonds. So, important to note here, proteins are held together by peptide bonds. A peptide bond is the covalent bond specifically between the amino group and a carboxyl group. So one amino group, through a dehydration synthesis reaction, forms a peptide bond, a covalent bond, by interacting with a carboxyl group of another amino acid. So here we see three amino acids. All three of these were featured in the last video as examples of different subtypes of amino acids. And we're just going to see a couple peptide bonds form here. Notice it's forming again through dehydration synthesis, where water is removed as the covalent bond is formed. If you look on here, we're looking at two peptide bonds between three amino acids. So we have a tripeptide structure, three amino acids joined to each other by two peptide bonds. It's common for a string of amino acids to be referred to as a polypeptide, many peptide bonds being present. For our purposes in this course, and this is fairly common among a lot of molecular and cellular biologists, we're going to use the term polypeptide to refer to the chain of amino acids, but we're not going to use it as a strict synonym for protein. A protein is going to be the functional final product, and we're going to see what I mean by final product in the next few videos. The polypeptide is the chain we begin with. So again, proteins are polymers of amino acids, and individual amino acids are joined to one another by covalent bonds called peptide bonds. In the next 5 videos, we're going to explore the concept of protein shape or conformation and look individually at each of the 4 commonly viewed levels of protein conformation.